also known as the isotropic band over here. So you have the A band, you have the I band over here, I band, isotropic band. Now look at this. In a sarcomere, you have an A band in the middle, and you have half of an I band over here and half of an I band over here. So one sarcomere is actually half of an I band, let's just clear this out over here. Yeah, let's just take that out. So you have half of an I band plus one A band plus half of an I band located. So this gives you one sarcomere. Do you understand? Now if we look at this, you have the Z line and the Z line here. This is the sarcomere, so you have half of an I band. Don't do the algebraic sum and say one I band and one A band makes a sarcomere. It's not that way. Alright, you have half of an I band, one A band, and half of an I band. The remaining half of this I band is in the other sarcomere, the adjacent one, and same over here. This I band, half of it is here, half of it is here in this sarcomere. So you get two different bands over here. Alright, so, and one more thing to talk about is there's a part of the A band where there is no thin filament overlapping. So that part we call the H zone. H zone, H stands for heller. Heller means light in German. Don't mistake it for light band, okay? Just say heller means it looks a little lighter in the middle, because there are only thick filaments over there. So there's a little light in the darkness. Okay, think of it that way. This is your H zone. So this is the organization of your sarcomere. And when you contract the muscle, the myosins are going to make cross bridges where the actins bind to them, and then they're going to pull the actins towards the middle. So you get these cross bridge formations, and the actin moves to the middle over here. The thin filaments move to the middle, and the Z lines move together. So can you guess which bands are going to change their length and which bands are not going to change their length? So if you didn't guess it by now, the A band is going to be exactly the same in length. It's not going to change. Why? Because it's where the actin and the myosin is overlapping. Myosin isn't moving. It's just going to bind to actin and pull it closer. So its reach is not going to change. But what's going to change is the Z lines coming closer. So the I bands will shrink. So remember, when you contract the muscle, the I bands will shrink. And remember that the thin filament is moving closer together. So these H zones are going to get smaller. So that means the I band and the H zone is going to get small, and the entire length of the sarcomere will get small, but the A band will be the same. So these I band and H zone will shorten, and the A band will stay the same during contraction. This is very important to remember, okay? It's an important point in your exam. Let's just talk about the proteins that again are present in these different parts of the sarcomere. The M line contains another protein called myomycin. Just be aware of this name, myomycin. Okay, so it forms part of the structure of the protein over here, and from the M line you get myosin that comes out over there. And if you look at the Z line, that's where you get the thick filaments coming out. The thin filaments. Z line is thin filaments, and that means actin is coming out from the Z lines on both sides. Now let's just talk about actin for a minute. And actin is a polymer. Okay, it's a polymer. You get these proteins called G-actin or globular actin. Globular. And the globular actins all will polymerize in along this one lengthwise, end to end, and you get what we call filamentous actin or F-actin. So the globular actins all come together and you get filamentous actin. So they all join end to end, end to end, end to end like this over here. Okay, and this is in an alpha helical configuration, and if you look at these actins, they have this little black dot I've drawn here. This is your myosin binding site, where the myosin will bind to the actin and pull the Z lines close to each other. Now, these actin thin filaments are not just going to be one single chain of actin. Actually, you're going to get two of these chains of actin. So it's going to be an intertwined, almost like a DNA double helical alpha, helical structure intertwined. So it's not a single fiber, it's double, like the one I've drawn over here. Can you see the arrow pointing over here? So you get these double ones. So I've drawn one which is in black and in alpha helical configuration, and you have the red one, that's the other chain of the F actin, which is intertwined over there. So that's how you get your thin filaments. They're made of actin and they are intertwined with each other. Now, when we look at this actin, it is also covered by another protein in the thin filament, and I've drawn it in green, and this is known as tropomyosin. Right? So you got your tropomyosin. So what is your tropomyosin? Well, tropomyosin is like, it's a protein, it's a dimer, it's two proteins stuck end to end. So this is one which I've drawn in green. This is a tropomyosin, and there's another tropomyosin. Okay, I've just simplified the diagram. I've removed one of the alpha chains, one of the actin chains over here. Don't worry about it. The thing I need you to understand is, when you look at actin, myosin loves to bind to the actin binding sites. It can't resist actin. If it sees that binding site, it will bind to it. Think of it like Romeo and Juliet, where myosin is Romeo waiting at the balcony, waiting to climb up the balcony, and Juliet is the girl in the balcony, or then myosin wants to steal a kiss from her every time. So what happens is, sometimes you don't want the muscle to contract, and that's when you want the muscle to relax. So what we need is, we don't want myosin to always stick to the actin over there. So to prevent the myosin from sticking to the actin too much, we have this protein called tropomyosin, which covers the binding sites for myosin. So these binding sites which are present on the actin are covered, sort of like a veil on Juliet's face. So myosin can't recognize. So is Romeo cannot recognize, is that Juliet or not? Knowing Romeo, he'll still take a chance actually. So what happens is, he would still try and climb the balcony, but nonetheless the tropomyosin inhibits the myosin molecule from coming and sticking to the actin molecules. So this is one important thing. Tropomyosin is present on the thin filaments, and they inhibit the myosin from binding to the sites over there. Now you have troponin also. Now troponin is another protein. Actually, it's three protein segments stuck together. So we have troponin T, I, and C. Okay, T is bound immediately to the troponin, to the tropomyosin. So this is troponin T. This red color is T over here, which is bound to the green that you see over here, the tropomyosin. So it's connecting them. Okay, that's what it does. Then you have troponin I. I is inhibitory to myosin even coming close to the actin molecule. What it does is, I want you to think of, okay, so I told you that tropomyosin is like a veil on Juliet's face, meaning the actin's face. Now, sometimes it's not enough to just have a veil. You need to look scary. So it's not just a veil. It's like those Call of Duty balaclavas with the scary face that you get, something like Tokyo Ghoul from anime. So you wear a scary mask. You don't just cover the face, you cover the face with a scary mask. So myosin, or who we call Romeo over here, doesn't even get the idea to come close. Like, no, that face is too scary. It can't be my Juliet. So this is your troponin I. It inhibits the myosin from coming anywhere close to the thin filament. And then you have troponin C. Troponin C is very important because it's where 4 calcium ions will bind, and what it does is, when the 4 calcium ions bind, they cause movement of the tropomyosin away. It slides away from the binding sites, allowing myosin to finally find the place to bind. This happens during contraction of the muscle. So when you cause contraction of a muscle, you make an action potential in the muscle, and that muscle will cause the calcium levels to rise in the cytoplasm. And I'll tell you how it happens. There's a sequence of events. The point is, you stimulate the muscle and calcium gets released into the cytoplasm, and that calcium has to go to troponin C, and that's going to move the tropomyosin, and that is going to expose the binding sites, and then myosin can bind, make the cross bridge, and pull along the thin filaments along to the close to the M line over there, shortening the muscle. Alright, so this is your troponin complex over here. Then you have your myosin. Myosin, if you see, is a hexamer. It's got there are 6 myosins actually, pointing in 6 different directions over here. Anyway, let's just go through a structure. So in our muscle, we have myosin type 2. Alright, there are different types of myosin, like myosin 4 is in your white blood cells that help in moving of the blood cells, catching bacteria and other stuff like that, microbes and stuff. So the myosin, I'm just drawing one section over here. This is myosin 2 molecule, which you could see. It's got a single chain over here. This is the tail of the myosin. It's got a nice long tail, and then it's got a hinge, like a neck region over there. And then you have the head. This is the head over here. So you have a head, a hinge, and a tail. So you have the tail, the hinge, and the head. So this is one molecule, but like your actin, you have 2 of these intertwined, as you could see over here in this picture on the right. So what happens is, I'm just drawing one over here. I've just drawn one here. This is the heavy chain of myosin, okay? What happens is you have a heavy chain, you have 2 heavy chains and 4 light chains. So each heavy chain at the head has got like 2 light chains, which are drawn in red over here. So totally you've got 6 different proteins if you look over here, and we'll just look at one of these right now. This is the heavy chain of myosin, and what happens is the head is what binds to the thin filaments, and if it binds to the thin filament, it does the cross bridge cycling. It's able to change its angle. You see, the head has an angle of around 90 degrees when it's resting or cocked. It's not when it's not contracting the muscle, but once the binding sites are exposed on actin, then what happens is the head will bind to this, and it's going to bend the head. It's going to bend over there, and it's going to go from 90 degrees to 45 degrees. So 90 is going to move towards 45 degrees. So the head will move from this position to this position, which is 45 degrees. Okay, so this pulls the Z line along itself, around 15 nanometers, I think, for each cross bridge cycling. This head movement where it attaches and pulls is called power stroke, and we'll be talking about this again in more detail. Just be aware of this term called power stroke. Power stroke. So power stroke is how the myosin, pulls the Z lines together by binding to the thin filaments. So here we have two myosin heavy chains intertwined, and there's an ATP binding site also near the head region. So this power stroke requires energy, and for that you need ATP to bind to it and to undergo hydrolysis, and gives it its energy to convert to bend the head from 90 degrees to 45 degrees, approximating the different Z lines together. So here in this diagram you see that there are 2 myosins heads over here, and there's the ATP binding site over here, and there are 2 of these proteins over here attached, also 2 chains. This is 2 myosin regulatory chains over here, and 2 essential chains, 2 myosin essential chains.